All right, well, good morning, everyone. Thanks for joining us today. I'm feeling privileged that under the circumstances, looks like we have gotten a pretty large group of people together, perhaps more folks than would have had a chance to tune into this normally. So thank you for your time, and I hope you find it well spent. So I served as a project manager for a COSSEP project that was titled Bioassessment of Non-Perennial Streams Affected by Oil and Gas Extraction. So a brief overview of what I'll be talking about today. I'll give you some background on the need for dry stream assessment in general, and also why that was of interest to the Office of Spill Prevention and Response. We'll go over the selection of indicators that went into this method. We'll discuss our site selection. I'll give you a brief description of the protocol, and then share the initial data that we got from this pilot project and some potential next steps that may be taken. Before I get too far into this, I do want to say that today I am essentially in a lucky opportunity to be presenting the work of others. Myself and Pete Odie were Office of Spill Prevention and Response staff that collaborated on the project design and its application to the COSSEP program. But the field work and data interpretation and species ID were conducted by a research team from California State University of Monterey and the Southern California Coastal Watershed Research Project, or SQUIRP. John Olson and Rafael Mazur were the PIs for the project, and I want to give a big thank you to Dr. Pete Odie at the Department's Water Pollution Control Lab, who helped put me together with these folks and helped coordinate the work with me. The need for bioassessment. Generally speaking, humans in industrialized countries have had a very significant impact on freshwater ecosystems, which is probably something that goes unsaid for this group. One of the best ways to understand the impacts is to monitor the relationship between the biota and the conditions of the above-ground freshwater network. Often, this same type of information is sought using chemical and toxicological testing, but in some applications that can miss the whole story. Using chemical and toxicology data requires that we have specific target compounds in mind that can be measured in a laboratory. Often this involves extrapolating from model organisms to the organisms that we're looking at in my needs for a spill. And we can have interactions among multiple contaminants. This becomes particularly problematic if we're looking at streams where we don't have a lot of model organisms that have been studied, or if we're talking about sensitive species where toxicology testing is unlikely to have been done. This need for bioassessment certainly extends to dry waterways. Additionally, bioassessments of freshwater have included flora and macrofauna within or near flowing stream beds. But low precipitation rates in very large areas of the western U.S. means that much of that fauna, and I'm really talking about fish and benthic macroinvertebrates, are absent, or in some cases all of the time. And this means that the health of these non-perennial streams, and when I say non-perennial streams, I'm including both ephemeral or intermittent streams, that meaning streams that are occasionally do not flow, or streams which are ephemeral that flow only when there is ongoing precipitation. In each case, it can be impossible to assess these streams during times of low flow or no flow. That leaves us needing a method that can be adaptable to intermittent streams outside the typical field season, or ephemeral streams all of the time. To scope the level of this issue, non-perennial streams make up 73% of California's streams. Based on the National Hydrogeography dataset, that includes over 200,000 kilometers of California's stream network that we are without an assessment method. And obviously, depending on the region you're in, that percentage of streams will change. So the image on the right there is one way of looking at this. This is California catchments that are predicted to be need this episodic method at all times, and blue streams there are riverine systems that could be monitored with a more typical bioassessment some times of the year. And the picture on the left is a breakdown of the non-perennial versus perennial streams by region. So the gray percentages there, those are the non-perennial streams in each ecoregion. So dry stream assessment is a larger issue than just the concerns of OSPR and spill prevention and response, but OSPR does have some particular needs where this method may be useful. Certainly, dry waterways dominate the inland landscape. Much of California's oil production occurs in areas with low precipitation. Our field response teams can have a need to monitor post-spill recovery and the efficacy of field methods. They often find themselves in the position where they need to help make a choice of how clean does a site need to be for the biota to recover as best as possible, and trade-offs of the impacts of those spill methods versus natural recovery are difficult to determine without data. I work in the Natural Resource Damage Assessment, or NRDA unit, and for us, we need a quantitative measurement of injury to scale the damages that we pursue in court. This means we need to quantitatively measure severity of impacts, the recovery times from those impacts, and we need a means to monitor the restoration that is put into place. The basis for developing this method is off of past work. Most similar work is occurring in Europe and Australia, where bioassessment of dry streams is more common. CSU Monterey, who we contracted with for this project, has been developing methods to look at dry waterways. They started with a project funded by the Regional Water Quality Control Board out of San Diego, and they also have a larger ongoing EPA project across California and Arizona, which we'll talk a little bit more about later. Essentially, this comes down to modifications of the surface water ambient monitoring program protocols for perennial streams, and the compositional and functional characteristics of terrestrial arthropods and bryophyte assemblages were our primary data source. One question that brought up, at least for me, is why the use of assemblages. The composition of a biological assemblage, because of the diversity of stressor tolerances, can offer a way to quantify changes in stream composition and contamination. This gives you a broader image than just looking at an indicator species, because we're using a full suite. In aggregate, arthropod or bryophyte assemblages can provide a broad spectrum picture, which is really what we're looking for here, is something that can be used in the vast majority of cases, not just those where we happen to have the ideal indicator present. Typically, when used for bioassessment, assemblages are characterized with a multi-metric index, where different metrics are used to describe different aspects of the community structure and function. For this study, we were focusing on terrestrial arthropods and bryophytes. So this top left picture here is showing what the pitfall traps look like used for collecting the terrestrial arthropods, and on the right is an image of a bryophyte, which is pretty similar to a moss. These are mosses that are present in very dry areas. These are good potential indicators based on prior studies, and I'll show you some of those reasons in a moment. The protocols can be readily adapted from other bioassessment protocols looking for benthic macroinvertebrates and plant assemblages, and the logistical requirements are fairly low. So here's an image from that San Diego study that I previously mentioned, where they looked at potential assemblages that could be studied. Some of the things they evaluate are whether or not these occur across the whole ecoregion, for obvious reasons, whether or not these are going to be present during a dry phase, if there was past studies that show that they respond to alterations, and if they had previously been used as an indicator. Also pretty critical is the level of diversity, since we're expecting to focus on assemblages and the changes in measurements like species richness. You need to have a really diverse number of organisms present. And finally, we need a relatively quickly deployable field method, so logistical requirements become key. And if you look through this matrix here, some of the best options are terrestrial arthropods, riparian vegetation, bryophytes, and potentially diatoms, but there wasn't a lot of information on whether or not those were going to be consistently present during the dry phase. They may be in the hypereoic zone, but since that's currently an unknown, we're not there yet. Our study focused on terrestrial arthropods and bryophytes. I would like to point out that the larger EPA method is also taking into account riparian vegetation with a desert ecosystem modified CRAM. Our project was not able to fund that for these sites, but it was actually done by the researchers so that they could include this into the larger EPA study. So some more detail on those other method efforts that I mentioned. This grew out of a previous study by the Water Control Board that funded a project developing and applying an MMI to San Diego County. And that was the source for the methodology that was used here. EPA has also funded that larger two-state project, which is sampling 100 sites, or did, from 2016 to 2019 across California and Arizona. It was my understanding they're pretty far through the data analysis for that project, and we'll talk about it a little bit more later. This may become useful to us. And there are also some other smaller targeted ongoing efforts starting up this summer to look at coastal non-perennial waterways and Central Valley waterways that are impacted by agriculture. Our project, however, was designed to be specific to Osprey's needs where we could. So that means we needed data focused on impacts related to oil production. We needed methods that would be quantitative to meet the needs of NRDA, methods that can be deployed quickly, and so those logistics and training requirements became key. It has to be something that we can use for this concept of but for the spill. Sometimes for general use of bioassessment, what you're really looking at is what is the overall health of this waterway compared to an average waterway. This is something that OSPR could potentially do to look at spills, but you would have to have a very large data set to be able to do this defensively. So but for the spill, for the purposes here, means a method that we could deploy with an upstream, downstream type control and say, what would this stream's assemblages look like without the spill, knowing that there may be other impacts, and look at just the change caused by spills. And finally, a correlation with oil concentrations. This comes out of our damages need to be linked to Limpert-King and the OPA laws, and so a strong correlation with directly to oil concentrations is important for us. Growing out of those needs, here were our study objectives: to characterize arthropod and bryophyte assemblages in dry streams along a gradient of increasing disturbance from oil extraction activities, examine how the stress related to that oil production affects the health of the dry stream, evaluate the ability of the dry stream assessment protocols to detect ecosystem alterations, and evaluate the usefulness of an existing MMI, and here I'm talking about the MMI trained from that San Diego study, to quantify damages associated with, impacts from oil extraction. So in my mind going into this, essentially this is a pilot study. And the key question for us was, are we going to get a strong enough signal from oil-related impacts that we can see a defensible change in these assemblages? And if we can see that strong biological response in the waterway, then we have a potential tool going forward. Once we had those general objectives outset, and that was the point of the proposal within, Pete, Odie, and I worked with the PIs on selection of our sites. Prior to selection, each site was evaluated for physical habitat, stream morphology, and human activity levels. This was done largely with GIS data. And so what we're looking for here is sites that are spread out, some on the eastern and western sides of the valley, but they need to have similar physical habitat, so you're in the same vegetation type, and as much as possible similar elevations, similar sizes of streams, and then selecting a gradient of human activity levels. And so for our 13 sites, this means 4 sites that, based on this precursory evaluation, appear to be minimally impacted. They don't have a lot of upstream in their watersheds in this image human activity. 4 sites where we expected to see impacts. And so for this initial study, we wanted a gradient of the level of impact, but we did steer towards relatively heavily impacted sites because it was really important to us that we see a strong signal, and it was anticipated that if this was going to move forward, that we would have to explore the full gradient of the level of impact later. And we did have one site, which was a previously investigated spill location where we knew the outcome. This was a buried petroleum site where there was a fair amount of manual excavation to remove oil. So from here, I'm going to walk you through the sampling methods that occurred in the field. So once on site, sampling begins with a physical habitat assessment. And as I go through this, I'll say now, this is essentially a 2-day protocol for each site. So the sampling begins with a physical habitat assessment. This includes setting out 9 transects 20 meters apart over 160 meter reach. And they're measuring channel width, depth, pebble count, and channel slope. They characterize the stream morphology by estimating percent cover of each microhabitat, looking at the percent of the channel that's currently wetted, and the percent cover of vegetation types. Once they have that physical habitat data, they also assess human activity based on the proximity from the field. This would be a lot to read, so I'll just tell you this is a list of the human activities that are assessed at each site. This is the same human activities that are evaluated for all of the EPA study and the San Diego study as well. These include land use, like the level of urbanization, chemical stressors, like known agricultural or industrial water contributions, hydrologic stressors like modification, physical stressors such as sediment disturbance or hardened features, and biological stressors like grazing or invasive plants. The arthropods are sampled using a modified pit fault trap. These traps are set at 8 random locations between each transect. So you're using a random number generator, and then that bottom left image there shows that random number between 1 and 10 is used to place the trap based on a percentage of channel width and the length of the transect. The ramps are set parallel with the direction of flow, and the traps are fairly simple. It's essentially a food container with some standard size openings and aluminum ramps. They're sprayed with a spray adhesive and coated with native substrate. And then propylene glycol is used both as an attractant and a kill agent. And the traps are left for approximately 24 hours. Essentially, this protocol can be used for 2 or 3 sites a day, where a field crew is going out, doing their physical habitat, and setting traps on the first day, going through their 2 or 3 sites for the day, and returning to each site 24 hours later to do their arthropod collections. The sampling methods also include collecting riparian vegetation associated arthropods. This uses a beating sheet method. So beating sheets are a relatively simple concept, but highly efficient, where you're wrapping a piece of vegetation with a seat, beating that sheet with a stick or a PVC pipe in this case, and then collecting the arthropods that'll end up coated onto that sheet. One of the difficulties with that method is getting a standardized size piece of vegetation. So one modification for this study was that they're using a cloth bag so that the amount of vegetation that can be fit into that bag is more standard. And doing this provides a better estimate of the species composition than using a funnel or a backpack operated vacuum. Finally, let's talk, but not least, certainly talk about bryophytes. So bryophytes are collected using timed searches. When the physical habitat is occurring, typically one person is doing that while the second field person is walking up down each channel, each side of the channel, and looking for mesohabitats. So, for example, looking for microhabitats, for example, a tree that has a root system in the stream or a large rock with a sediment deposit, and identifying those, and then breaking the microhabitats up to a specific amount of time search. So we have a 12-minute search. If you had located 6 suitable microhabitats. You would search each microhabitat for 2 minutes, and the microhabitats are then sampled for that allotted time. The equipment here is rather simple. Best tool that I saw used was essentially a spoon. Each bryophyte is collected and enclosed in a separate envelope. And don't have an image for you here, but the amazing thing to me, being non-trained and going out with this crew, is your first walkthrough. They let me do this without providing the best tool, going up and down and seeing what I could find, and I didn't do all that well. But the expert's tool is a spray bottle. If you see underneath the vegetation there's that green color to the bryophytes. These organisms, when they are provided with a little bit of moisture, will respond to that moisture by greening up, and in some cases opening flowers. It makes them much easier to find very quickly. And finally, we also sampled hydrocarbons. The hydrocarbons were collected as soil samples using a 5-centimeter metal sampling ring. The crew selects a part of the channel with deposited sediment, hammers a ring into a 5-centimeter depth, and then digs around that ring with a spade. These were stored in a glass jar, shipped off to SQUIRP for a total petroleum hydrocarbon analysis. The TPH was measured using freeze-dried soil samples. That helps to preserve some of the volatile components. So once those samples are collected, the arthropod and bryophyte identification follows some months later after the field season. Arthropods are separated into trap caught and vegetation caught, and then identified to the family and morphospecies. Generally, this corresponds to identification to the genera. The sorted dry bryophyte samples are separated by morphospecies using a dissecting scope, and then identified to the family or genus based on leaf morphology using compound microscopes. One thing of note, arthropod analysis seems to be something that many laboratories can learn to do. This bryophyte identification, at least thus far, is a pretty niche skill set that requires quite a lot of experience. For the needs of OSPR, in addition to those other human stresses that we mentioned, oil-related stress was also evaluated. This was done using well density based on Department of Conservation data, percent oil field area in the watershed. This was used by matching up the oil field polygon and the watershed polygon. Log oil concentration coming from the SQUIRP hydrocarbon analysis. Median sediment size, which was calculated using the pebble size counts from the physical habitat assessment, and percent fine sediment, which was also collected using that pebble size count. The metric analysis and MMI scores were adapted from those commonly used in traditional bioassessment. So we're talking about species richness, taxonomic composition, diversity, and feeding groups. The richness was evaluated on multiple levels, order, family, and morphospecies. And the MMI average scores were calculated using the MMI trained from the San Diego study. So let's move into our data. Here is the correlation between oil field stressors. So here we're just comparing those 5 oil field stresses to the log oil concentration. I largely wanted to point out here that one of our strongest correlation is between the oil concentration from the sediment and the percent fine sediment. And so this correlation is part of why when I talk about the data, we're essentially treating sedimentation as an oil field related stressor. I am aware there are certainly other plausible causes of sedimentation, but in the areas we were working, by and large, all of the human activities are related to oil extraction. So our arthropod results. Our traps caught a total of about 4,000 individual arthropods. We were pretty happy with this. Going into the study, this trapping protocol hadn't been used in an area quite this dry, so we were pleased to see that the trap counts were pretty good. Saw good species richness that ranged from 14 to 24 taxa. Arachnids were the most common. Also saw a lot of springtail and ants occurring in the traps. The vegetation-associated arthropods saw quite a lot there as well. I'd say part of that is that the crew is essentially choosing the one best piece of habitat for that bag on each site. Here, the species richness was much more variable. This was a more noisy data set looking at the vegetation results. And true bugs and or weaver spiders were the most common taxa collected in those vegetation. Many of the arthropod metrics responded to the measures of oil-related stress, and beetle metrics were among the most responsive. So here are the top arthropod metrics for each stressor. So trap Caloptera, that's beetle abundance relative to oil concentration. Trap Caloptera formicadea, that's beetles and ants richness to percent oil field cover, Beetle log abundance to oil well density, beetle log abundance to percent vine sediment, and beetle and ant richness to median sediment size. So the R-square values here were pretty strong. The red dot there is our one known spill site. And I'll talk about that in a little bit, but we were pleased here to see a very strong response, particularly from the beetles correlating with oil stress. Bryophytes also went well. Here's our main points for bryophyte results. Bryophytes were collected at 12 of the 13 sites. Going into this, there was a little bit of concern about how many bryophytes would we realistically find in an area this arid. And we were pleasantly surprised to see just how many we did find. Image on the right there is from the Berry petroleum site that I mentioned. And a total of 122 samples were collected from those sites. The samples included 20 different morphospecies. Brachia were the top indicator of oil and sediment stressors. In general, brachia richness had a positive relationship with increasing stress. And so I'm really glad you asked that question, Kathy. With the bryophytes, we saw a lot of bryophytes richness increasing in response to increased oil field stress. So here are the top bryophyte metrics. So brachia richness compared to log oil concentration, bank brachia richness relative to percent oil field cover, bank, and I'm going to butcher this one, Podiaceae richness relative to oil well density, and brachia richness relative to median sediment size. And so almost all of these bryophyte metrics, we're seeing increased richness relative to the amount of oil field related stress. Once these samples were worked up, they were compared to the multi-metric index that was calibrated in San Diego. And this MMI did not distinguish between the most and least stressed sites. So while we saw this really strong, well-correlated response from both the arthropods and the bryophytes to oil field stress, we're not seeing a good response out of the multi-metric index. And there are some potential reasons for that. One is that the trap beetle relative abundance was an increasing metric in the MMI. So the more stress they had in these San Diego systems, the more beetle abundance they saw. But that was a very strong negative correlation in this area. Interestingly, the Berry petroleum site was the only known spill site of the 13 sites and had the highest MMI score using the San Diego metric. I do want to point out here that the oil concentrations at the Berry petroleum site were lower than average compared to the other sites in the study. And so, as I mentioned, I think at the beginning, that site, the residual oil was largely excavated and removed from the system. And it looks like the cleanup technique did a pretty good job of pulling the petroleum contamination out of that soil. A lot of the sites we were at have historic oil concentrations that we were expecting to see. So we'll talk a little bit more about this MMI not calibrating well in a moment. But I want to move on to the overall question for us from OSPR's perspective was feasibility. So we saw a strong biological oil stress, response to oil stress. Many metrics had a correlation with oil stress gradient. I only showed you the top 5, particularly those metrics that related to beetles. But that MMI correlation was pretty poor. And some known impact sites, that Berry petroleum site and a couple of others where we were expecting to see our highest impacts, really didn't trend with the MMI. Talking to the PIs about this, there are some not unusual reasons for this to be an issue. The habitats may have responded differently because the average level of stress in eastern arid San Diego County and these oil fields that we selected are pretty different. So if you use areas with a very high degree of stress, that being this data set, and compare it to something with a relatively low amount of stress, those correlations can be problematic. Also, this area is much more arid. And the San Diego study and this study are both considered pretty small for calibrating an MMI. So the San Diego study, I believe, was 30 sites. Our study was about 13. Typically, when constructing an MMI, you want to use something on the order of 50 to 100 sites. And so we may just have a shortage of data. So again, just to summarize, the arthropod response to oil field stress, beetles showed a negative response to increasing stress. This is similar to what's been seen in previous studies of beetles, that they can be useful bioindicators and also respond negatively. They also respond negatively to grazing and urbanization. Beetles and other sediment-sensitive invertebrates will respond negatively to increased disturbance. The fine sediment deposition that we saw may contribute. And this strong relationship between arthropods and sediment was not observed in the San Diego area. And this is possibly due to stronger effects or hydrologic modification and chemical alteration that we're seeing because of the level of urbanization there. And to summarize the Bryophyte response, Brachia showed a positive response to increasing stress. High moss richness has been associated with intermediate levels of disturbance in other work. Increasing richness may be caused by the formation of gaps that allow new colonization of previously excluded species of bryophytes. If the level of wastewater disturbance is at a desirable level for brachia and other mosses, it could explain why we're seeing this increase in richness with more oil field activity. This could be due to moisture. It could also be due to nutrients or change in sediment stability. Once bryophytes colonize an area, they're pretty stable until they're acted on by something. And so this kind of formation of gaps could be crucial. So having seen that data, here are some of the future possibilities we've been thinking about with this. The results, particularly that strong correlation with beetles, suggest that a quantitative impact assessment is plausible, particularly if we're talking about fine sediments and oil deposition. Oil and gas extraction may have a stronger effect on invertebrate and bryophyte assemblages than urbanization or agricultural stress in San Diego. And that really is our hypothesis as to why that San Diego MMI was not ideal. In order to get a more robust result using an MMI, essentially we would need to recalibrate that index. And there's a couple of ways we could look into doing that. One is to calibrate the index with a much larger sampling. Essentially, this can be done by collaborating with the EPA effort that's collecting 100 samples across 2 states, and includes some much more stressed sites, particularly sites that have a high degree of impact from mining and extraction activities in Arizona. We could also consider collecting a more representative sampling of oil stress gradient to allow a regional MMI. You may recall when I talked about selecting sites, our first priority was seeing, do we get a strong response that suggests this method could go forward for OSPR's needs? And so we picked almost all sites with a high degree of stress. And if we were going to try to construct an MMI specific to, say, the San Joaquin Valley area, we would need to have a training data set that also included sites with a lesser degree of stress. And that would require a much larger effort, but we could do that targeted in the San Joaquin Valley if it really appears that the types of stress that we're seeing and the type of biology that we're seeing doesn't respond with the same trends as that larger effort. And with that, I'd just like to say thank you and let you guys have this photo of this little guy that I found on my day playing in field with the crew. Thank you. Thank you again to our collaborators who really did the heavy lifting on this, and thank you to the COSSEP program for funding the project.
So if the slope's going high to the left, down to the right, is that That's a good correlation, or what is that? Does that mean something, the direction of those lines?
So yeah, if the slope is going left to right, that's a negative trend. So if the slope is falling left hand to right side, that means, for example, the top left graph, that the abundance of beetles is falling with increased oil concentration.
Okay.
So then the bottom right graph, which is beetle and ant richness to sediment size, is the opposite. So as the median sediment size is increasing, the richness is higher. And you'll see on this and a similar side for bryocytes that some of these metrics are dropping in response to stress and some of them are increasing. And I'll talk about that a little bit in the discussion, but that is important. But one of the nice things about using a multi-metric index is that, biology, some things in a biological system may do well in response to stress, things that move in quickly or things that are more tolerant. Because they're doing better doesn't mean the system is healthier. And having a multi-metric index can help, give you a one number index of stream health and, leave kind of going into what benefits from stress and what doesn't, so when you have to have that conversation. I believe that was Kathy. Kathy, did I answer that question for you?
Yes, thanks.
By CSU Monterey Bay, there is a graduate student who I believe may have finished now, who did a dissertation on bryophytes, and I believe he still works for the laboratory and does their identification. Part of EPA's two-state effort includes them providing some seed funds to try to attract additional laboratories to do that analysis. So ours was done by CSU Monterey. I'm not aware of another laboratory that can do the bryophyte work in California now, but I'm hoping they'll be online soon. And WPCL has talked about maybe moving into doing some of the arthropod analysis if this project continues as an internal project, but I don't recall, and if someone from WPCL is online, correct me if I'm wrong, I don't think they're considering doing the bryophytes at this time. Kathy Bruce later asked, do we know what eats beetles in these regions? Obviously, it will depend on the particular beetle, but yes, they are a broadly used food source. Blunt-nosed leopard lizards certainly eat beetles. Some kangaroo rat species prey on insects, which I assume includes beetles, and bird species make use of beetles as a food source. Alan Sow asked, given that the bryophyte increases in richness with the increase in oil production, what does this mean for resource injury? That one is, I think, going to be a little bit longer answer. It could mean a few things. One thing that came up when I submitted the second COSSEP proposal was that some folks said, well, we don't want to be in the business where we're going to have to provide credit in the injury assessment for increasing the level of bryophytes. And that's part of why I was talking about kind of the benefits of a multi-metric index. I don't think, in my opinion, and this could change with more data, that we would want to do a bryophyte-specific analysis because of the difficulty explaining that increase. But in something like a perennial stream, looking at benthic macroinvertebrates, this concept has become more familiar. I mean, we're used to looking at an index based on EPT taxa, where some taxa are considered sensitive and some are considered pollution tolerant. And so when we're looking at richness, we may see an increase in the relative richness of those pollution tolerant species. And we don't consider that good for the health of that stream. We consider that an indicator. And so I think having that increase in richness with bryophytes could help contribute to a multi-metric index given enough data. I think if we try to focus too much on what I would maybe poorly call a pure toxicological study, where if we were going to try to say, well, you killed bryophytes or you killed beetles, it would probably not help us. I'm going to hope that answers the question. I talked around it a little bit.
Are there any other questions that anyone has? Okay. Well, thanks a lot, Dan. Again, we really appreciate this.